meters to go.
See you coming into
all right, folks, we are pretty close to the bottom. We are at 1,875 meters from the seafloor of Horizon Guyot.
Altitude.
My name is Chris Kelly.
Meters.
I'm with the University of Hawaii, and I'm serving as one of the two scientist co-leads on this particular cruise. And right next to me
Let me know
is...
you'd like me to go into auto
This is
heading.
Chris Ma, and I'm an expert in sea stars from the National Museum of Natural History in Washington, D.C. at the Smithsonian
I got you
Institution.
tight, just barely there.
So we are inside the Johnston Atoll Unit of the Pacific Remote Islands Marine National Monument. So we are inside a monument. We are at the northern side of the monument on a seamount called Horizon Guyot. It's a very, very large seamount, and it's got some very cool aspects to it. One little fun fact is that it is the very northernmost seamount in the Line Islands Seamount Trail or Seamount Chain, which extends more than 4,000 kilometers all the way down through Palmyra, Kingman, Jarvis, and it continues way on down south below that as well. So we are at the very tip. So that's kind of very interesting. And the other fun fact is that Horizon has... there was a few dives on the other side of Horizon many, many years ago, but it's such a large seamount and it's virtually unexplored. So we are here today, and we're hoping to try to find
the edge
some
of
things
fisheye
about this seamount, and I'm very excited. We tried to get on this in 2015, and timing and logistics didn't work, and so we're giving it another shot. And I'm looking forward to finding out what type of animals that live on this place.
And there are certainly a lot of animals to be expected. Sea cucumbers, corals, sponges, all of the wonderful things that you've come to expect from watching us in the past.
My main Zeus
Some
is looking
remarkable
back as far
things
as I
that I
can.
hope we'll see. Sea cucumbers that swim, sponges made of glass, and corals that stand 3 to 5 feet tall. It's 1.5 meters tall sometimes. So all sorts of things that I hope we'll encounter.
A couple things for those of you who haven't been on the Okeanos website now. This expedition or this particular cruise is called Laulima Oka Moana, and it basically is using Hawaiian words in reference to partnerships. Because this type of project is very, very collaborative. In fact, when we have an opportunity, we'll introduce the ROV team in front of us. They're from one group, and I'll let them describe what they do. We also have OER folks on board, NOAA folks. We have monument folks who are attending here. All of you who are in the event log, we've got a whole bunch of people in the event log. I'll call them out later.
That'll get us down.
I'm from University of Hawaii. Chris is from the Smithsonian. So it is a very, very collaborative type of endeavor, and that's one of the reasons why we decided to name this cruise this way.
290 maybe.
Okay.
And so we're slowly descending like that monkey song, going down.
Is maybe 330.
I'm comfortable with this to start on down.
Okay. I'll bring my rotator like 45 or something.
Pushing out.
So I'm going to go ahead. I don't know who's on the actual audio line here, but right now we have 5, 6. It looks like at least 20 people on the event log, including Asako Matsumoto. She's from the Chiba Institute in Japan, and she's a premier octocoral biologist who's participated on many cruises so far. Got Derek Sutcliffe, Ken Sulak, a fish biologist. Good to see you on the event log, Ken. Les Watling, another premier deepwater coral specialist at the University of Hawaii. Mike Ford, midwater specialist and expert, who's been, no doubt, paying very close attention on the way down. And here we go. The bottom is in view. And I will introduce other participants once we get settled out here a little bit.
Just checking my Z bias here.
I could see a few animals already, I think, unless they're just shadows.
Pretty good delta. I'm trying to catch up.
Yeah, backing off the Z bias.
There's something. Yeah.
I'm thinking those are crinoids on top of some kind of barren stock, perhaps a sponge stock.
Keep her coming.
Crinoids are called feather stars.
No, that's okay. No,
They're echinoderms. We'll talk about more. We'll talk more about that in a little while.
Okay, got a 16-meter delta, 22-meter
yeah,
altitude.
so we can see some animals already, so this is a good start. We're relatively close to the summit of this seamount, by the way. The summit actually starts at somewhere around 1,900 meters, so we're just barely below it.
I think we'll keep our bailout plan of 065.
We've got a bunch of animals. We've actually got what appears to be a bamboo coral just going off screen on the left, as well as a glass sponge, that kind of tall thing that's now in the left lower corner. Crinoids, Chris already pointed these out, and these look like crinoids and possibly another type of bamboo coral. I'm not really sure.
I think it's safe to get them out.
I agree.
We have this very interesting stock structure with a lot of tentacular extensions coming off of it to the left. Perhaps a sponge?
Oh, yeah. Is this...
What is that?
Well, it could be an extremely tall Walteria, but that's taller than any Walteria glass sponge I've ever seen before. So we got a bunch of stuff to zoom in on, and when they all settle out and they've got everything ready to go, which they'll let us know.
Thank you, Bridge.
When we can start taking some close-ups. So, Chris, do you think these crinoids might be in that genus Glyptometra? The kind of, it's in the family Trinometra, if I remember correctly.
I have your lowers partially deployed, as you can see.
That is something that you have me at a disadvantage for. I'll have to double check.
I'm just going to tow them out like that. That's all right. Yeah.
Is our starboard shelf a little inboard, or is that always in the camera view now?
I can check that. Starboard wing outboard.
So typically, when we begin our expedition like this, the ROV has to settle out on the bottom and orient itself. The pilots and engineers have to calibrate and
bring the slider pretty far away from
sort of settle out the ROV before it can begin proper operations along the bottom. And that's what we're often waiting for in the initial stages of our exploration on the deep. So just hang on tight, and we will begin the descriptive part of what we're doing shortly.
In the meantime, let me mention a couple other people that are in the event. We've got Cole Morgan, who's been a regular on some of these dives as well. We've got looks like Nicholas. Okay, sorry, she's sitting next to me actually. Nolan Barrett. Nolan, when the names are all merged together as a single name, I read it a little wrong. Nolan Barrett has also participated on some of our expeditions. Scott France is a regular; he's on there, and Tim Shank. A lot of people know Tim from Woods Hole, and Tina Molodtsova from the Shirshov Institute in Moscow. So this is great. We appreciate all the help we can get in figuring out what all these animals are that we're seeing.
Too low.
That's all right. You're all right. It makes us look good.
Copy that. Not too much heaving today.
Co-pilot,
indeed, and as a new participant, I'd also like to welcome all the people in the chat room. And just to comment that we have scientists from multiple places throughout the world. Tina is from the Shirshov Institute in Russia,
let you know
as well, and we have people from Japan, as well as people from all over the United States operating with us shoreside. Ooh, looks like there's a glass sponge in the center of our view.
I'm going to make sure your drawer's all the way
So we're going to try to annotate this video as best we can. Nicola is actually being very helpful in that respect. She's from Hilo in Hawaii as well, and she's on Seascribe, as are some of the rest of you. This particular glass sponge, I'm pretty sure, is something that we've seen quite a few times before. It's in the genus Tritopleura, and that's one thing I know a little bit about sponges, because I'm really fascinated by them. I know enough to be dangerous, let's put it this way. So I'm fairly comfortable with that ID. It looks like we have a primnoid coral that's going off screen right now, and this could be something in the genus Calyptrophora, possibly Calyptrophora angularis.
Interestingly, over to the left, it looks like there's is it a coral or an Antipatherian with little aplacophorans whorled around the base. They're little orange curls. Is that an ophiuroid or are those aplecopherans? Aplecopherans are small worm-like mollusks.
Okay, video.
Yeah, I'm not sure.
Toe hold. We can get a little bit of
Stay tuned.
Yeah, I think they're just settling in, get some zooms here.
Yes.
Going to come left.
So we're barely on the bottom, and already we're seeing a multitude of animal diversity. Brittle stars.
We can continue to zoom.
Yep, that's what they are, and this coral is indeed in the family Primnoidae, and I'm trying to see if we're going to get some help in the event log. I sort of called it a possibly Calyptrophora angularis.
Yeah, what are you thinking
I'm
for
seeing if
direction of
anyone
movement?
wants to come up with a counter ID on that.
And I think your identification of Wealth area looks pretty close for the sponge.
I was trying to plan ahead a little bit.
Okay, if we can go macro just for a moment.
So,
we can see the,
Scott is pointing out the bamboo coral, and yes, we are most definitely going to image that bamboo coral after we get this primnoid.
Okay, thank you, Bob.
And the
let's come out a little
is this in a...
yeah, these look like members of the family Ophiacanthidae.
Yeah.
And this is a widely occurring group of brittle stars which lives on the surfaces of bamboos and other deep-sea cnidarians. They're commonly encountered, but they're known for having really sharp spines.
Yeah, so Pilot, if we could get a zoom in on that coral that's coming up from the bottom of the screen right now.
Okay.
And then after that, we'll get that black coral in the background, but this white one that's coming up is different than the one you imaged a moment ago. This is going to be a bamboo coral, I'm pretty sure.
Chris, I'm hearing you, but not through the intercom.
Sorry.
So you need to press the...
Okay, yeah, sorry about that.
I gotcha. I gotcha.
So you read my mind,
yes,
Dave, and so Dave's the pilot today right now, and so yeah, we got a request to get some zoom in on this bamboo.
Okay, let's go in video. We'll get macro this.
So for those of us,
Scott French here. And
welcome, Scott. Please tell us what we're looking at.
yeah, I was just having a look in the database to compare to some of the things that we saw back in 2015 when we were down here in the Johnston Atoll, and I do believe that this is similar to what we called at the time a Jasonisis bamboo coral. So, I mean, we're looking at a planar octocoral fan, a sea fan, and you can see it's very white, and you see some intermittent dark nodes. Those nodes are arising where the branches are arising. That black is protein, the white is calcium carbonate, and there's tissue over it. So we called this a Jasonisis, but we were managed to collect
Do you want lasers, or I guess we haven't had lasers on yet?
Genetics on the piece that was collected, we determined that it was
yeah, when we pull out.
Chris, I know that Kelly, I know that's something that you are interested in, keeping those sorts of notes. So I suspect that that is the same. And in 2015, we collected this at 1,981 meters up in Papahanaumokuakea. So pretty much the same depth. Interesting to see it down here.
Okay.
Great, thank you very much. And that was the voice of Scott Franz from University of Louisiana at Lafayette, who is another octocoral specialist, and he does genetics on bamboos and chrysogorgids and black corals and all types of things. So thank you very much, Scott.
So for those of you who are just tuning in, these are a certain type of coral. These are animals, not plants, and they are distinguished by having these feeding polyps, all of these little sort of flower-shaped structures. Those are feeding arms that encircle a mouth right at the center. This is a colonial animal, and so all of these polyps contribute by feeding food to the rest of the body. They're often called bamboo corals because if we look closely at the structure, the sort of stalk below them, they're white with dark bands at specific branching points, hence giving them a superficial appearance to bamboo. And if you denude the tissue, if you take the tissue and all of the feeding soft parts off, that's what you see, is black and white bamboo-like porcelain type of skeleton. They often are quite large.
So, pilot, when we're done with this bamboo, there is a black coral that was sort of above it and beyond it just a little bit in the background, and I'm pretty sure Tina Molitsova would love a good look at that one if we can see it.
Okay, video, let's come wide and find out where we are.
There he is.
Okay.
There it is right there.
Let's zoom from here. We can reposition, but let's get a look at it from here. We're stable. And hold there if you would, please, Bob, and just get a little focus. There's some... Okay, what I'm going to do is reposition for that. Come wide again, please, Bob. And is there anything in this scene from our present perch that you'd like to see closely before I move over there?
Yeah, pilot, I think we've probably covered this particular perspective.
Okay. Off we go.
Okay.
Chris, I know that you had noted the whip with the long, what appeared to be tentacles coming when you sat down, and I'm going to assume that you will have a close look, but I just wanted to be sure that we do indeed get a chance to see that.
Yeah, I agree, Scott. That's on the list here. I think getting this black coral is sort of on the way. Yeah, it is, because this whip with the long extension is just beyond that. So maybe we'll hit them in a row here, get to the black, and then if Dave the pilot can get over there and get that one, which I'll bet he can, then we'll get that next.
Ooh, there's a nice sponge over onto the right side there.
Happy there?
Yeah.
Yeah, there
for a
is.
move pretty soon here. I got him at 26.
I believe that Waltheria-like sponge, but with a brittle star hanging off the tip.
Yeah, maybe a sponge, but Scott's also wondering whether this could be a bamboo with sweeper tentacles, so that's why there's a couple possibilities
do a short move
for it. I think it's going to come out to be a Waltheria sponge, Scott, but we'll find out in a few minutes after we get this black coral image.
15 meters, or is that too much?
So those of you who are going black coral,
15.
for those of you going black coral, what the heck is he talking about? That isn't black, it's orange, for crying out loud. Well,
for crying out loud, Chris.
So it's the skeleton there. The actual tissue is in black, but the skeleton underneath it is proteinaceous and has a very black, beautiful texture to it.
So if I'm reading Tina Molitsova from the Shirshov Oceanographic Institute in Moscow, she's calling this one Chrysopathes. And so a lot of what we do, a lot of the identifications, are made based on cooperation with scientists all around the world. So thank you, Tina, for assistance with identification there.
Yeah, I think Tina is, possibly her comment is that she would love to potentially consider this for collection. I don't know, maybe we can take a little poll here. Tina, do you think this is something new, or this is something you've seen? Is this similar to other Chrysopathes that we've imaged on the trip? The trip being the capstone project for 3 years. It's a pretty long trip.
Are you feeling tugs for me? You want me to try to give you some more slack, Dave?
I think Tina's just asking for the
I see it
close-up
getting
of the
tight.
collection.
Oh, okay. All right. Yeah.
Going to the base here.
Okay.
Thank you, Rich.
So now you're seeing why they're called black corals. You can see the skeleton that's a bit denuded of polyps right there where it attaches to the substrate, and certainly has a black look to it. There are these little crabs. Well, they're not really crabs. I guess they're squat lobsters that often are found on black corals, and often very specific species associated with the very specific black corals.
Yes, Antipatherians have a very interesting skeletal, or they have a interesting substance covering their stalk. It's actually composed of chitin, and it forms very particular weaves along the surface. The name Antipatherian, it means against disease, and they were originally believed to have medicinal properties. And I don't believe that's ever been shown. I could be wrong. Perhaps our resident experts could clarify on that. But they're very distinctive deep sea they're members of the group of the phylum Cnidaria. They have stinging cells, and we'll see them as we progress along the bottom.
Video, push focus far. There's something waving from that other. Oh, okay, yes. It's much less intriguing when it's in focus. Thanks, Bob.
So we're probably good for science if we want to then go to our next target, which would be that either sponge or weird coral behind this thing.
Okay, wide, please, Bob.
You got a little bit of telly left, Dave. We're moving your way.
Okay.
So.
Sponge in the far distance, or on the left. Here we go.
I agree with you, Chris, that this looks like it's going to be a sponge, but it's different from any of the other Walteria I've seen.
Yeah, this is a really tall one, Scott, and it's got very long lateralia, the lateral projections, I guess, that are coming out of the side of it. He's going to get a close-up, and we'll be able to potentially go a little bit further than this genus. But a Walteria is a glass sponge that's in the family Euplectellidae, and the glass sponges have got these marvelous, beautiful microscopic parts that are called spicules that give it some structure to it, and the taxonomy is based a lot on these microscopic structures. So over the years, these sponges have been collected and examined, and then we're starting to match video images to the taxonomic identification of a lot of these animals, so we're starting to be able to identify them more and more just from video. So this particular one is probably something close to Walteria leucardi, and so we often refer to this as Walteria CF leucardi. And what I'm looking at is that center core area, and it's got these round holes in it. And this is basically places where the framework, the spicule framework of the sponge, the scaffolding, there's been silica that's actually been added to that scaffolding, and it's starting to fill in all the holes between this marvelous scaffolding, and it's created these round openings where water could still get into the sponge. You can see some brittle stars that are growing on it right here.
Forces are very light.
Copy.
Yeah, they're kind of living in and around, and kind of, I don't know if they're inside or outside, Chris.
Yeah, it looks like they're actually inside. Sometimes they can just crawl in when they're smaller, and they probably just take advantage of the water currents. I would also like to just point out to people that, so this is a sponge, this is an actual animal, and the body structure is actually composed of glass, and so all of these projections that you're seeing are essentially sort of living silicon oxide extensions of this animal. So sometimes it's miraculous to think that here's an animal that it's almost like it's out of science fiction. It certainly looks like it's from out of science fiction, but these projections are all made out of glass, and there are animals that live inside them, taking advantage of the water currents. These are brittle stars. They're related to sea stars, and they're probably have their arms projecting outwards and using their spines to take advantage of small organic particles in the water as the currents bring them by. I've also been informed by Nolan Barrett that Anthropitherians have, in some instances, had bioactive sterols isolated from them. So the medicinal use thing is not entirely without some evidence. And sometimes these glass sponges can be quite large. They're obviously very intricate and very beautiful. But a few years ago, when Chris was surveying I believe it was the northern Hawaiian Islands, we found a glass sponge that was the size of a Volkswagen bus. It was in all the papers. It was remarkable. Sponges and a lot of the other corals that we're seeing here are sometimes referred to as ecosystem engineers, and that's because their very bodies essentially create habitat for other animals. And so what we're seeing here is not only brittle stars, but these squat lobsters, and perhaps these browner animals that are living along the sponge are hydroids, if I'm not mistaken. And so the structure of these animals actually provides habitat for more animals, and thus more animals. And so they are very important to the ecosystems in the deep.
We're good when video's good, pilot.
Let's come wide, Bob, if you're got enough in the can there.
I moved toward you, so we got some tether to play with.
Very well. Okay.
I'm lined up on 330.
That should be along the contours.
I think that's pretty much the direction of movement. So
yeah, we got one last item in the neighborhood, I believe. This sponge out here.
Yeah, so we're headed to this larger sponge over here to the right, and since Chris actually spotted it, and just confirming what it is, and yeah, I can already identify it to genus, at least from here. These are very common in the deep below 1,000 meters for sure. This is in the genus Poliopogon and the family Pheronematidae. So the pheronematids in general a lot of them tend to be very massive sponges. So in some cases you can see these things that look like Jabba the Hutt down there. They're really that quite big. And there's another sponge right next to it, pilot.
Yes.
And maybe we could go for a twofer here before we go. But this massive one is attached to the rock via spicules. Very loose spicules. It's not glued to the rock, and those of you who have been following the Okeanos cruises hear me talk about this quite a bit, because that's part of identifying its what species it is, or at least what genus it is, by how it attaches to the substrate. So you got two sponges that show completely different morphologies. They're both glass sponges, and I've been talking about the one on the right, the Poliopogon. And this one over here to the left, I'm pretty sure from this distance it's going to be something called Pherea in the genus Pherea, but I'll wait on that until we get a little bit closer look. And then maybe after this we'll get going a little bit.
Science. Let's get a quick zoom here, Bob, and get a look at that structure.
And then there's the Anthomastus in the lower right-hand corner.
That's right. Excuse me. There was an Anthomastus, so we can get a quick snap zoom of that as well.
Okay, you can pull out.
One of the really neat things about the sponge, the Poliopogon sponge, is if you look at the base, all of those fibers that connect it to the bottom, those are reminiscent of fiber optic cable. They look very much like it, and they feel very much like it in texture, including if you accidentally pick one up, they can give you splinters. Oh, little isopods on that one.
There's something inside too. A little shadow of.
Chris, I'll follow up your comment by noting that there have been some engineering studies of some of the giant spicules of the monorapis glass sponges, deep-sea glass sponges that grow in these very long single stalks, and they've found that they can transmit light better than human-generated fiber optic cable.
Wow, better than. That's amazing.
That's pretty amazing. Yeah, thanks, Scott, for that. This particular sponge, I'm reasonably confident it is in the genus Pherea, and it's something we call Pherea near Oka erecta. There's two forms of the sponge that's been identified as Pherea oca. One of them is kind of a bushy format, and the other is kind of a columnar, standing up kind of morphology, it tends to have a more erect morphology, and that's why we call it the erect form. I'm pretty sure that's what that was. But
So I'm heading up the ridge here.
Oops, so we missed that Anthomastus, right? Well, we'll hopefully see another one.
No,
sorry, Tina.
Yeah,
I can go anywhere you like.
Oh, no, that's okay. I was down by the big massive sponge that we just imaged. And I was looking up the ID of something here.
I can get right back there if it's...
I still see that large sponge in one of your manips.
Yeah, it's right over here.
Yeah, we're pretty close, so maybe it'd be worth a quick look at that. We've got Tina on
maybe the red one,
the line here, who's expert on these Anthomastus. Yeah, there's the sponge
lower
right there.
right of the big one.
And it's to the right and just below it, a little bit further below the ROV, I think.
You have no red at this range.
I remember. I saw it out of the corner of my eye. I was confused about what the object of interest was.
Yeah.
But
there's a
I
sea
see
cucumber.
it right there.
Yeah, that's the guy hiding behind the rock.
Oh, okay.
We are down and stable. Clear to zoom video.
So we're going to wait on Tina's ID on this because she really knows very well. And she's been teaching a bunch of us for the last few cruises, or more, or last few years,
That'd be okay
I should
for
say.
a 15-meter move after this guy?
Sort of how to ID these things, but we certainly don't have the eye that she does.
Or do you want 30 meters or 15 probably?
You know, why don't you...
You could always put more in, but...
Probably should make a pretty good jump here, otherwise we'll end up smelling the flowers too long. And
okay. I think 330 is still a good direction.
Okay.
So yeah, we'll call it a 30.
I think that's good pilot when video's good on this one, and that certainly shows Tina what she needs to know.
Okay, thanks. Yeah, call it in.
Yeah.
Tina identifies this as pseudo-Anthomastus. Thank you, Tina.
Like another move, please. 30 meters, bearing 330 degrees, speed 0.2 knots. Good copy.
So these kind of substrates that are sort of mixed are always visually appealing to me because they have a really nice, I don't know, something about the lighting which is really beautiful, plus the fact that we can get an assortment of animals.
On the right side, next to that, there's a yellow crinoid. It's a feather on a stalk next to the white sponge. It looks kind of like a glove. The sponge looks like a glove. And so within the course of, we've been down here only half an hour, we're already seeing the third of the 5 classes of echinoderms. So this looks like, this is what's called a stalked crinoid. This is a filter feeding animal related to sea stars and other echinoderms such as sea cucumbers. what these animals do is essentially they hold their arms up into the water, and they can capture particles with these extended projections, and they move caught food into the mouth, which is right at the center. And these are immobile. They're sessile. They don't move on their own. This is one in the family, I believe, Hyocrinidae, possibly Hyocrinus. These kinds of animals are found in the fossil record as far back as the Paleozoic. So that's some 600 million years ago, before the time of the dinosaurs. The video is really nice because it shows the small feather-like projections coming off of each of the arms. And I think we even saw a little, there's a snail that's on one of the arms there. Eventually when we get around to the other side, there's a family of snails called the Eulimidae, and they often will parasitize these animals and try and exploit their bodily fluids.
So, Chris, this particular stock crinoid has got those ring structures just below the calyces, and we collected a couple of these, and this is a newly described genus. And for the life of me, is it the Chuck Messing, who's a crinoid specialist, actually told me what this is. Maybe I've got it. Give me a moment. We'll figure it out. I got it. Whew. All right. So it's a newly described genus called Tiburonocrinus.
Oh, neat. Tiburonic, the ones from the North Pacific.
Yeah.
That's probably named after the Tiburon, the Mbari research vessel.
Yeah, I believe it was.
One of my first jobs when I crossed into ocean science was to design some components of the Tiburon.
Oh, wow. So this is coming full circle.
Yeah.
So sometimes you can see like anal structures protruding from around that oral region. That's where a lot of times the excrement vents out.
See if I can swing around here and line us up for a better view.
Oh, wow, that's beautiful. Yeah, you can, if you look closely, you can see that little white, I think it's a snail. Yeah. And that little white snail is, like I said earlier, is probably a eulimid, and it's probably got its proboscis. It's got a tube stuck into the body of this crinoid and is taking advantage of its bodily fluids. Feeding, in essence. But wow, you can see the fine hairs along the surface. These are what the animal uses to help filter food out of the water.
So, Dave, we have a request to look down the stock a little bit before we leave this crinoid.
Sure.
And just sort of maybe pan down. I think Tina might have spotted something on the stock a little further down. And nope, looks pretty clean. Maybe something right at the base, but I can't really see. Oh, it's just something that's right in front of the base, that's all. Okay, yeah, that's good for science whenever video is done on this, Dave. I think
yeah, why, please, Bob. It's about time for me to take a little bump up the hill here. Yeah. My co-pilot's catching up to me in anticipation of adventures ahead.
Thank you, Bridge.
Looks like I got.
And as we're cruising over the substrate, some of you may have noticed that it's got a very bumpy texture to it, these rocks. These rocks aren't naturally bumpy. What has happened is the surface of the rocks have been coated by what we refer to as manganese crust over the course of millions of years. And I'll be quiet here because I'm looking at these corals here, but maybe we'll make a little bit of distance and then we'll zoom in. I have a feeling we're going to see animals all day here. This is certainly a good start.
I'll try to give you a good view in passing here.
Yeah, with all the sediment, oh, that could be a highland matted sponge that we just went over. And we may see some sea pens because we've got an awful lot of sediment, and these are some cnidarians specialized in living in sediment. Keeping my eye out for those. There's another Walteria sponge that we already imaged earlier on.
What's that in the distance there? See if we can scoot over there. There's something on the same rock as a glass sponge, lower center.
Looks like it's probably another crinoid.
And then this thing in the
do we have lasers on?
Center. We can turn the lasers on,
thank
yes.
I'd like to see how tall that sponge is.
You got lasers.
Okay, there's our 10 centimeter reference.
So to those watching, oftentimes it's very difficult to reference how large any of the things we're seeing with the video are. And so we have this guide, these two lasers, which are, I believe, 4 centimeters? 10 centimeters apart. And they give us a guide as to how large the things that we're seeing are. And so this is
meter plus.
I know about a
co-pilot, rotate the lasers, please.
We'll get right on that.
Yeah.
Yeah, I guess also it'd be about a meter.
When you'll be able to go back through the clip, and with the spots on the base of the rock, you'll be properly scaled to step it across the image.
Yeah, that's fine, pilot. We can go ahead and
just to give everybody a sense of how big that it was, it's about you said about a meter.
Yeah, that's what you're saying too.
So about a meter, so that's like 3 feet tall. So that's something that is the size of like a very big suitcase. That is a tall column made of glass. So glass sponges are always a favorite of mine.
Thank you. That's good.
And so what we have here is a feather star, also called a crinoid. And crinoids, as I mentioned with the stalked type earlier, they take advantage of water currents. And so what a lot of there are two different forms that we'll see, one of them with the stalks and this other type without. And they find themselves at the top of very tall structures in order to put themselves into water currents higher up above the bottom. And so we'll often see them perched on dead sponge stalks, corals,
and other
was
the same one we saw earlier, the Glyptometra. So it's,
yeah.
I'm not sure we need to annotate it again. One thing that would be good is if we noted every species that was new on here.
More of the same.
A little bit of tether left.
Yeah, you might have got another
Should
move
be about
going.
done swinging if you get moving again.
Yeah, we need another one going here. And what are we?
Same as last move. So
Okay,
good.
so I'm
330, yeah.
just right up this nose here.
All right, I'll call it in. Bridge, nav. We'd like to repeat the last move. That's range 30 meters, bearing 330 degrees, speed 0.2 knots. Good copy.
So, as an overview, this is D2 looking north, due north. View, 5 meters above the bottom. Depth of 1,907. Coming left now.
For those of you who might just be tuning in, this is the Okeanos Explorer on our 2017 expedition to Johnston Atoll, the Laulima Okwa Moana expedition. The name represents or translates as Partnership for the Ocean, an effort that is an important element of our research. Cooperation is an important aspect of all research, and our expedition is no different. We're currently on Horizon Guyot at about 2,000 meters or so, and we're slowly traversing the bottom, exploring this unexplored region of the Johnston Atoll area.
This is the view looking due west, 270 degrees from D2. 5 meters off bottom, 1,908 meters. Appears the axis, local axis of the ridge is northwest. And pushing ahead. Set structure there.
Something down there. I think the pilot has spotted something on this boulder, this rock right here. Looks like a black coral.
It's actually
a little bit different species.
Fascinated by the thing to the left there.
I think this is a dead sponge stalk that's fallen over.
Okay.
And
all right. Looked unusual.
Yeah. So before we head up, maybe we could take a look at this black coral.
Sure.
Looks like it. I'm going to go ahead and let Tina weigh in on this. Yeah. And then we will relay. She's on the event log, and we'll relay her comments to those of you who are listening to this broadcast.
You know, it's interesting, Chris. There's a lot of these dead sponge stalks in this area.
Clear to zoom video.
Okay, so Tina is slightly delayed on her video feed, so we're going to give her a little bit of time to get caught up and get a good look at this coral. So one thing as she
has a little clay horizon just to its right of the black coral on the rock. Oh, you're already zoomed in now. Sorry, I'm delayed. But when you zoom out, you'll see there's a little stalk to the right on the edge of the rock.
Okay, yeah, thanks, Scott. Yeah, appreciate it. Well, and the pilot heard that, so he'll get a.
So we're about halfway through the move. Nav, would you agree?
About that, yes.
So Tina went ahead and actually logged this for us in C-Scribe. This is great as a Bathopathes in the genus Bathopathes. So thank you very much, Tina.
Quite yet, maybe.
Bathopathes is fairly. Yeah, there's the little thing I think Scott was talking about.
Oh wow, that's interesting. This is another one of those little mystery animals, Chris.
It could be. I don't know what this is, quite frankly.
It has interesting segmentation, yeah, and it looks vaguely bryozoan-like, but maybe not. Do any of our experts on shoreside have any notions about what this might be?
Well, I can say I apologize for pointing out something so small, and I can't see the resolution I have either. anything more than what I offered before.
Yeah, it could be a Cladorhiza. It's got, there's a bunch of things that we had tentatively ID'd as bryozoans, and I sent off all the pictures of things that could be bryozoans to, I've forgotten his name. I've got to remember his name. It's somewhere on my list here. But anyway, of the various pictures we sent him, he said only one of them was a bryozoan, and he didn't know he had a variety of different potential phyla for the other, for all the other images. So there are, of course, plenty of mystery animals down here that we encounter. And
let's come wide, Bob. It's time I was moving on.
So Tina says, no, not Cladorhiza. So that's a Cladorhiza is a kind of sponge. And Dougal asks, if that was full zoom?
Yes.
And yes, Dougal, that was full zoom.
Yeah, Scott thought it was a bryozoan. That's what I certainly would have thought until we got some feedback on some of these pictures. But this is a little bit different, and some of the things I sent him pictures of had branches on them. This one didn't have very many branches, so this may have been something different.
Remember, we're looking for rocks too, right? Anything large and detached.
Yes.
Anyway,
sort of a interesting bunch of crinoids on dead stalks. Again, these climb up in order to take advantage of the water currents that are passing through that area. Oftentimes there's an optimal current that they exploit in order to feed in the best possible way. And so it has to be not quite so slow, not quite so fast. It has to be just right. And so these distances are often the best for filter feeding.
As a general observation over this substrate, it's interesting that the density isn't particularly high. I mean, actually, right now we're seeing quite a few things, but there seem to be more dead sponges than live sponges so far.
That's very interesting. I was noticing that earlier. The sponges, there are a lot of down sponges, and a lot of these sponge stalks that are projecting above the surface are dead. That's interesting.
And there's something squishy there.
Yeah, these are a couple more. Oh, I see. Yeah, let's go ahead and get one of these sea cucumbers.
Okay.
Since we happen to have sitting right next to me one of the foremost echinoderm experts on the planet. So what the heck? Let's zoom in echinoderm. What do you?
Sure. Let's give it a whirl.
And Chris sits very closely at the Smithsonian to Dr. David Pawson, who is also a specialist in these sea cucumbers, and of course Tina Molitsova and at the Shirshov Institute of Moscow has got a colleague there, Andrei Gabruk, who's also a very well-known sea cucumber expert. So
clear to zoom video.
Okay, so of course our first sea cucumber has to be something controversial.
Yeah.
Let's put it this way.
I think this is the one that goes back and forth between being it's a synallactid, I believe, and it has that sort of hem or that fringe around the edge, and so it's unclear exactly which genus, but it is close to something like oh, and now the name blanks. That's even better.
Hansenothuria.
Hansenothuria. Thank you.
Yeah, and so Andrei Gabruk, when he looked at some of the pictures of this beast that I sent him a couple months ago, he had no problem sticking with synallactid and possible Hansenothuria. Synallactids tends to be very, very transparent sea cucumbers, and Hansenothuria has very small to almost absent, I guess, a pseudopodia on the back. You don't see very many of them on here. Dave Pawson came back, and he was looking at kind of the skirt that's around the bottom edge of this sea cucumber, and he thought that was more in line with a genus called Benthodides in a very different family. So it's, but then Benthodides are typically not this transparent. So it's,
Yeah. This is often the difficulty with studying animals like this, is we depend on their external features to tell them apart, and oftentimes when they're described either incompletely or that the animals themselves are simply very difficult to work with, and the structures are not very clear. So all of the fins, all of the different tentacles that we use to tell them apart might not be present in past specimens that were used to assign the names. This one is really also just interesting for most people who might not have seen a sea cucumber before. You'll note that the body is actually translucent, and that gray-brown kind of tube running through it, that's actually its gut. These feed on sediment, and so what they do is they ingest all of the fine organic material along with the sediment, and it goes into their gut. They digest the organic sediment and defecate the cleaned castings out the end. And sometimes we'll see those on the deep sea floor. But what you're seeing is literally right through the body wall and what it's been eating. So we'll see other examples where the body wall is quite clear, and we'll get to see almost the granule size of the sand or the sediment that they've ingested.
Let's come wide video. Thank you.
So we had a question from Tina about how thick these crusts are that we're looking at here, and this is this bubbly covering of all of the rocks down here that we've been looking at. And you know, there was actually assessment of the manganese crust down in the Johnston area that was done back in 1987. There was a report done when they were considering this area for possibly deep sea mining, and they had stated in that assessment that the crust in this area averaged around 5 centimeters in thickness. This particular seamount has been dated to between 88 to 90 million years old, so mid to late Cretaceous. So it's had quite a while to accumulate a pretty thick layer of this crusting on it, and so I think that's one of the reasons why this area was of interest for developing a mining industry a couple decades ago. Those reports, of course, were ahead of their time. The technology, of course, is not caught up, and the industry isn't really taken off yet, but people are getting very interested in mining the seafloor because this crust material, as well as nodules you find elsewhere, have very valuable minerals that at some point in the future we're going to run out of from terrestrial sources. And so it's probably a reality, and that's one of the priorities in the capstone campaign that we're doing in the Pacific, is trying to document who lives on crusts and actually lay the foundation for understanding the communities that could be impacted in the future of mining activities.
330.
Oh, wow. This is interesting. A nice little sediment pond.
Yeah, with some ripple marks in there, bed forms.
Huh.
This may be the top.
It looks like the water got a little murkier too for some reason. You notice that?
I'm a little high off the bottom right now. That accentuates. There's a bit of snow, fine snow, but when I rise, the contrast goes down.
Yeah, if you take a look at our camera 2 on online, there's a great view of the Sirius imaging the submersible, and it's traversing the contacts between the manganese rocky area that we've just been surveying and this flat sort of sandy bottom pool that's sort of distinctly present away from it. It's really quite fascinating.
Still good at 330?
From a sedimentary geological standpoint.
Bridge, nav.
Just saw a little anemone and possibly a little caliphate sponge on the bottom.
Bearing 330 degrees, speed 0.2 knots.
Animals have spread out a little bit here where we're at right now, but maybe because of the sediment that we're getting.
Good copy.
Yeah. On the next move cycle, let's bump them up to 60, 2 tether lengths, so that we're covering a little bit more ground.
Or we could move faster. yeah.
So again, for those of you who are new to the Okeanos dives, couple things. You'll see these little periods where the ROV is sort of sitting in place for a little while, and this is because it's extremely complicated to move this type of vehicle. You're basically moving three things: the ship, the Sirius vehicle that's up above, and that's being the video from the camera from that camera sled is being shown on feed 2, and then the ROV itself. So you got to have everything kind of lined up. And you start with the ship, and then you move things sort of in sequence, and that's how they get up and going. So that also offers some opportunity to do some close-ups. So pilot this little wire coral that's kind of sticking up off the side of the rock. I'll bet there's some interest in that. If we could take a look.
See if I can get scooted over there. I'm a little bit out ahead. Yeah. So maybe bobbing around, but we'll get close.
You can see a bunch of stuff in the background too, so maybe the density is going to pick up a bit as we move a little further upslope. It's a glass sponge that's going off screen on the left here. It's got one of these, what we think is a Glyptometra feather star on the top of it.
We have a request from Tina to go after the small white colony after the whip.
Got it. Okay. And that's the bright white thing. You think that's what she's talking about, or which one is
she? That one?
Yeah, I'm not sure which one she was referring to.
Small.
But let's go ahead and get the whip first.
Yeah, clear to zoom video.
We call these unbranched Gorgonian corals whips, for obvious reasons. This is a pretty interesting one. Excuse me, what was that, Scott?
I was wondering if from a distance it was a Candidella, a primnoid whip.
You know, I would agree with you now that we have the advantage of seeing the close-up before you do. Good call, Scott. It has a few more...
that the lower third of the colony doesn't have polyps, and that's really consistent with these Candidellas.
Yeah, this is that potential Candidella that's got 4 polyps per whorl. That is at least 4. Looks almost like 5 in some cases, yeah.
I see 5 at once.
The classic Candidella gigantea had 3 polyps per whorl, but we've been seeing a lot of variation in that, so I'm not sure it's a real fixed trait for them always to have 3. Or possibly it's a different species of Candidella.
So we got a nice shout out from Dougal Lindsay at Jamstack. Keep eyes out for small reddish benthic jellyfish attached to underside of overhangs, please. So that will be an interesting thing to keep an eye open for. Small red underhanging jellyfish.
Looks to me like this is 3, 4, or 5. Is that unusual to have a different number on the same colony?
Yeah, so we have seen that sometime. I know what you're talking about. There's 3 up near the tip, and it almost looks like there's perhaps more toward the base. So maybe as the tip is growing, there's fewer, and then they add polyps. I don't know. It's a good observation. I think that was. Not sure if that was Dave who made that comment, who I think is a navigator. Yeah, hi Dave.
Hi. We have dual Daves.
At some point you guys need to introduce yourself when you get a moment.
Do you know what our layback time is, Matt?
Okay, we had a request from Les Watling to put a laser dot on the axis.
Laser dot axis. Okay, let's come wide, Bob, and let me...
Thank you.
Yes. Now that dot is somewhat beyond. Let me put it down at the base, and then we can scale from that.
So those of you who might be concerned because we're lasing a coral here, these laser dots are just used for scaling. They can't possibly hurt the coral. They look bright to us, but they're pretty dim. They're actually the way that most vehicles are able to get some idea of sizes in the deep sea. They're very convenient. So again, those two dots are separated by 10 centimeters.
Chris, do you know what the diameter of the dots themselves are?
No.
That's totally dependent on distance.
Okay.
You know, they're not perfectly collimated, so they diverge a little bit.
Thank you.
I'm going to swing right to the...
2 to 3 millimeters, Dave, would you agree with that?
Yes.
Again, depending on...
So Scott saw something on the... I guess that's what...
That was
I'm hoping that that's what
requested, I
Tina
believe.
was...
Oh, this is interesting. Is that a coral on the...
it's a dead base, I think, of something. Whatever was here, it broke off, I think. And then it's got some hydroids and some other things growing over it. That would be my impression of what we're looking at.
There's a coral, I think, farther up.
Yeah, and then Scott thought he saw possibly some stoloniferous octocorals, or there's a cup coral, a single polyp hard coral, scleractinian.
What I saw, Chris, was a little hard to tell. There was a line. It looked like a yellow line, and it would be, when you zoom out, it would be, I think, to the left. And it may just have been a trick of distance, something about reflection from the rock surface, but it kind of reminded me of lines of stoloniferous octocorals or lines of barnacles that we've seen on other
Just come wide, please, Bob, just for a minute.
I remember I know what Scott's talking about.
Yeah, so Pilot, did you copy that?
Yeah,
that line.
Okay, back in, Bob. It's the squiggle. It's the object of interest, to use the technical
Sea
term.
squiggle.
And...
Oh, it's a stoloniferan. Way to go, Scott.
The base of a sponge that's broken off.
Scott, excellent eyes. It's a stoloniferan, I think, but we'll actually let you take a look at this and throw out your opinion since you spotted it.
Yeah, absolutely. Stoloniferous octocoral. So we've been seeing these sea fans, these octocoral colonies, but some species can also grow kind of like a ribbon, or it can grow in 2 dimensions, much like a mat. So this is a beautiful view where you can clearly see the ribbon of tissue that connects the different polyps and the polyps extending from them. They look fairly large, and maybe there is one stalked barnacle kind of hiding in amongst them. Nice.
Yeah, barnacle on the right.
Yeah, he's referring to that one, on the right of the screen, about a quarter of the way on the right of the screen. But anyway, excellent eyes. And what's interesting is that this is related. It's an octocoral like some of the branched ones, as Scott pointed out. So there are all types of ways to solve the problem of surviving down in the deep sea. You can have branches, you can have single whip colonies that we've looked at, and you can string yourself along the seafloor.
Oh, yes. Really one of the more unusual types of octocorals that we see down here. So octocorals are one of the most commonly encountered types of corals or cnidarian animals that we see. They're distinguished by the fact that they have 8 feeding arms around the mouth, hence the name octocoral.
Okay.
Let's come wide, please, video.
Bridge, nav.
I guess it's also worth noting that there have been some early hypotheses that perhaps this was a very simple form of octocoral and therefore might be, if not ancestral, at least more close to the ancestral type of whatever gave rise to all of the octocorals. But the genetic data don't support that the ribbon-growing forms are any less advanced than any of the branching forms. And if anything, they may be more derived, that is, evolutionarily, the ancestor to all the octocorals probably was branched or had a skeleton.
But not a drop to drink.
Yeah, evolution. I'm sorry, go on.
No, go ahead.
I was just going to say, evolution in the deep sea is a crazy thing. There's all sorts of different relationships, and what sometimes appears primitive is, there's a sea urchin down on the sandy bottom there. It's the
trails here.
Oh, yeah. Oh, baby.
And we also got some corals after this on the rocks that I think we haven't seen before. So again, we're seeing things basically every few feet on this fencing. Yeah, so that after you do the urchin, that sort of wispy coral there would be great.
There we go. Did we need a close-up on the urchin?
Could we look at the urchin?
We can look at the urchin.
I would appreciate that greatly, sir.
Yes. Whenever something excites you, speak quickly, and I will.
Yes.
I will act.
Okay.
And we've definitely got some new corals in the background that are coming up. The density is increasing, which is a good sign.
Still climbing slowly, right?
And so we're also seeing a lot of trails. Those are always informative for people who study these. You can actually find these types of trails in the fossil record as well from sea urchins. Oh, look at this. Oh, and a brittle star. It's like a ice cream echinoderm sundae. So this is, there are, sea urchins often occur in sort of two groups. The sort of more round, also known as the quote-unquote regular urchins, and the irregular urchins, which tend to be bilaterally symmetrical. That is, there's a right and a left side. And these actually dig actively through sediment. And this, I believe, is that kind of urchin, which we'll get to after we've quickly imaged this very stunning pink brittle star, which is very similar to the genus Ophiumuseum. I'm not sure if that's exactly it, but it's in the same family, the Ophiuridae. I'm sure my colleagues who study brittle stars are rolling their eyes at me right now.
I wonder if you can punch that name into C-Scribe, Chris, because you're the only one who knows that one.
Yeah, so
I don't think we're going to get anyone else entering that.
So right over there it is.
Right. Okay. And so what's happened? So the sea urchin is you're seeing all of these spines, and they use these spines to dig through the sediment, and they basically burrow through it. There's a mouth on the front, and they're just sort of ingesting the sediment and picking all of the organics out, and then similar in a fashion to the sea cucumbers, the sediment at the end is sort of excreted on the other end, and they create these really nice trails. But this one in particular has a really dazzling array of very sharp spines. The tests, which is to say the skeleton of these animals, tends to be extremely delicate and very thin, and oftentimes they're pretty much just suspended by or filled with water. But in samples of these kinds of animals that we have in museums, these fragments are so thin that you can shine light through them, and they're translucent. So as much as part of me, perhaps if we see one towards the end, we might attempt a collection, but collecting these would be extremely difficult and would be very similar to picking up an egg with a lot of sharp spines on it. So I think for now we're going to appreciate its aesthetic and look at these. I'm not sure, but I think they might be respiratory structures on the surface. But and it's sort of digging trail through the sediment.
So do you think this is in the family kind of 30?
Oh, no. These are irregular. I'm sorry. So these are what's called irregular urchins. So this is something like a spatangoid. A member of the group Spatangoida.
Wide, please, Bob.
And
boy, I blew that. I was showing the wrong picture here.
No.
He's got to log it.
Sorry,
no, it's fine. You're describing it. We just butchered the idea, or I should say I did.
No, I just need to make sure that's fine. And so those dig through. Did I? Oh yeah, there it is. Okay, yeah. So they basically dig through the sediment, and you can find representatives of sea urchins like this in the fossil record. They're quite common in some parts of Europe in the Cretaceous. But that particular one, I think, was sort of similar to something like Mauritia, which has a lot of spines that are projecting off like that. But I will leave that identification to my colleagues who know better. But that was definitely kind of an irregular urchin. Those are a really great example of how you have a derivation of a general body plan into a very derived one, because again these show a lot of specific adaptations for digging through sediment relative to the round sea urchins that you might be more familiar with in shallower water. The shallower water ones that occur on rocky bottoms tend to be round and pentaradial or radial in shape and symmetry. These, again, become bilateral. They have both a right and a left side, as well as the 5-part symmetry, and and that's partly associated with their ability to dig through this sediment. And at this depth, there are some very interesting examples of these kinds of sea urchins, and I suspect we'll be seeing their trails as we cross along the sediment bottom throughout the rest of the cruise. Some of these can attain really bizarre shapes. Some of them look like Coke bottles.
Little reminder for those of you who just joined us, we're on Horizon Guyot, and we are currently at 1,905 meters below the surface. Horizon Guyot is located in the Johnston Atoll Unit of the Pacific Remote Islands Marine National Monument. So we've been cruising just below the summit and probably coming up a little more on the summit because we're seeing more sedimentation. Sedimentation is pretty common on Guyot or flat top seamount tops because it's flat and it accumulates sediment. I'm just going to make a comment for those you might be interested, because we were talking about the difference in dissolved oxygen levels in this area versus the Northwest Hawaiian Islands. We actually hit the oxygen minimum at this seamount at approximately 600 to 650 meters depth, so a bit of shoaling compared to the Hawaiian Islands, where the dissolved minimum is normally hit at around 750 to 800 meters in the main Hawaiian Islands, and then even as deep as 1,000 meters up in the northwestern Hawaiian Islands. So it's a phenomenon we're taking a look at, where we're seeing essentially the oxygen minimum increasing in depth the further north you go, and decreasing in depth the further south you go. So if we, in Palmyra and Kingman, the lowest oxygen you get as you're doing a dive, it can be as shallow as 200 meters or so. So we do believe that oxygen is important for the communities down here. So we are definitely measuring oxygen and temperature and salinity and other environmentals as we're doing these dives, and hoping to get a feel for how these environmental parameters are affecting the communities we're observing.
Well, actually, this is maybe the first detached rock we've seen. I don't know if it's big enough or if there's any interest, but everything else has been cemented so far.
Yeah, roger that, Dave. So I think we're going to skip the rock collection today, because I think all we're going to find is manganese-covered sediment, given that we're so close to the summit. And and given if we had all the time in the world, if our dive wasn't a little bit shortened today, then yeah, we'd go ahead and grab some things. But I'm not certain we're going to get any valuable samples. But thank you for asking.
Fair enough.
It was the most interesting thing in a sea of mediocrity. I just had to stop. But it gets better here. You can see.
Yeah, that we've been looking at animals all day, and Dave's already saying that he's seeing not a whole lot, but we would like to see a little more diversity than we're seeing. We're seeing a bunch of stuff, and we've gotten some great images, and Chris has actually seen some of his beasts.
We saw a kick-ass batangoid. I don't know what you're bothered with.
Yeah, see, the sponges have been okay, but nothing out of the world, and the coral guys probably are. They'd probably like to see a few more corals than we've seen.
They saw stoloniferous octocoral.
So we have
doesn't get much better than that.
We have got a stock sponge coming up from the left here, Dave. I think you can see it's sort of waving. And this is at least something different.
Is this Bolosoma?
Maybe or maybe not. Look at that. He's showing off. He already probably knows what this is from that distance. He's been looking at the guide.
Hey, I've been watching this. There's people on Twitter who probably understand Bolosoma as well as I do.
Unfortunately, I'm not really sure it is Bolosoma.
Oh, well.
It was a good guess, though.
Yes.
What was the...
Thank you, Rich.
I think a better candidate for the stock sponge, which the pilot's coming up to right now and is going to zoom on, is something called the Calophacus. It's in the family Roselidae. It again is a glass sponge, as Chris pointed out. They're made of glass, silica, and they're really beautiful. They're deep water specialists. The shallowest glass sponge that we've documented in at least the central Pacific here is 350 meters down, so you're never going to see these things snorkeling or diving.
Clear to zoom video.
Yeah, so I think we're looking at Calophacus. Just like the corals and echinoderms and other animals, there's all types of solutions to surviving down in the deep ocean. We saw a massive sponge that was attached to the rock via all these fibers, and we saw a very transparent one with little tubes that we call Phoria, and then now we've got a stock sponge here that is in a very different family altogether. So that's my best guess on this one, Calophacus.
So I think, let me ask him.
Probably have this in the can, don't we, Bob? Come wide, please.
So Dr. Henry Ricewig up in Vancouver is working on this particular genus of sponges because they're not very well described and not very well documented. A lot of the members of this genus are known to subgenus, and you don't see that all that often in taxonomy. So the actual, perhaps best identification for that one animal is something that we call Calophacus subgenus Calophacus, and there's a variety of other types of subgenus as well. There's no species ID on it. It's probably an undescribed species that he's working on right now.
Hey, Chris, can I ask you a question?
Yes, you can, Scott. Please do.
So I don't have a good sense of how close we are to the edge of the Guyot, because I didn't see the revised shortened dive plan. So can you give me some sense? I mean, are we moving right along the edge? Are we 100 meters in, 200 meters in, more or less?
It's a good question, Scott, and I'm going to relay that to the navigator and see if he can give us a little bit of information about that. Watch lead 1 to nav.
Sure. Do you want me to answer that on OKEX?
Yeah, why don't you go ahead and answer that directly.
Okay. So we're about 50 meters to the east of what we think is the ridge, where the top rather, where it starts to flatten out. To our east, it's quite a bit steeper. To our west, it's flat, and we're maybe 10 meters below that flat section.
Clear to zoom video.
I hope you're that, Scott. I was so distracted looking at the sponge. But let's see. Let me see if I can see the screen. Oh, yeah, okay. So I'm sorry, Dave, you're saying we're right on the edge, is that correct? Pretty close to it?
I think we're about 10 meters below and 50 meters east of the edge, but it's sort of rounded out near the top. There's not a sharp transition between the slope and the flat top, or at least it doesn't appear like there is from the sonar.
But yeah, I agree. It looks like we're awfully close to it. Yeah, so we're pretty close, Scott. The edge is just not a really sharp even though this was a result of a landslide where we are right now, it's not a very sharp ledge. The sponge we're looking at right now is yet another one. It's a stock sponge. And this one is called Saccocalyx. It's in the genus Saccocalyx, and it could be pedunculatus as well, the species.
It was doing that a little bit
one of the things I'm noting to go to an ID here is all these lobes that are coming out of the side of it, plus that osculum on the very top part. It's sort of a very distinctive sponge. It's on a stalk as well. Very, very beautiful.
See what happens when I lift off, because I might be putting turbulence.
It's the Sirius one. Yours is
good. I know, but
okay.
I'm in the neighborhood. And wide, please, Bob.
I'll watch.
Yeah, we don't know how good the new one is yet.
Okay, we're continuing our survey here along this ridge.
This is the first cruise that it's been on the actual transceiver. Because we're showing pretty far to the northwest up there, right?
I'm surprised there aren't more sediment animals here. You know, the sediment does look thick enough. We actually saw some bed forms or ripples earlier on, which is suggestive of it being at least a few centimeters deep.
It might have track. You know what? You could try.
Well, I get the impression that some of them might just be buried.
It's off the map.
Something over here to the left. I don't know if we could get to that, pilot.
What's that?
There's something to the left.
That thing right down at the bottom?
Yeah, it's a black coral. There it is.
Tracking for a few minutes.
Oh, that guy.
Yeah, the fork thing there.
Yeah.
Wonder if we could take a quick look at that.
You're pretty far out there right now.
Yeah.
So Andy, just restart the software.
I would
not
just,
anything else.
And there's an interesting orange cup Idarian. Is it a coral or a zoanthid? Just south of it.
Should still be
we can zoom.
Do that.
Afterwards, you might think about putting another move
in. All right, Dina is in the event log. I think she's watching this dive, so we're going to wait and get her opinion about this. Her or Scott. Scott also knows the black coral pretty well.
Sure.
I have an idea. I'm not going to spout it off because I'll probably be wrong. But
another ship move, please. 60 meters, bearing 330 degrees, 0.2 knots.
Yeah, I'm getting a little tether tug here. I'm going to have to get going. Wide, please.
That's great. I think that'll be what she needs to help us with the ID.
Good copy.
Thank you very much.
And let's take a look at that sort of orange disc to the lower left.
Okay, I'm reposition.
Thank you.
Restarting. Good power cycle to Deucer.
It's still, it's worth a shot.
Okay.
Oh, there's two of them. I'll line up for both of them.
So not only Chris is interested in this, but Dougal Lindsay from Jamstick. And he's, I assume that he's watching from Japan right now as well. He was interested in those orange discs.
Okay, we can zoom. Here's the closer one. This will clear up in just a second.
Dougal Lindsay is a midwater animal specialist, but he also knows a lot about the benthic animals as well. So we're actually delighted to see him in the event log today and watching this dive. He, of course, would love to see a few more jellies than we've seen, but he's taking the cup corals at the moment. So that's what it looks like to me, a cup coral.
Yes, that's what I would say.
Yeah. And Dougal says he's watching from Yokosuka, just south of Yokohama in Japan.
Right.
Some of you may remember Dougal. He helped us out on a midwater, a bunch of midwater transects we ran. I think it was 1 or 2 cruises ago, and just did a fabulous job. He was really funny, so we're trying to get him watching more often and commenting.
Thanks, Bob.
He had some great comments, and he's extremely knowledgeable about the midwater animals. So another cup coral. This is hard coral. You're seeing the actual live tissue sort of draped over the hard skeleton, that white part, and the tissue is pretty transparent, so you can actually see the skeleton underneath it.
Right, so this is a proper coral, similar to the type you might see in a shallow water habitat.
Over here.
Instead of large colonial skeletons, though, these are single cups, a single animal. Imagine, if you will, a large colonial coral as sort of an apartment complex. This one is more of a single house, and it's living down here at the deep. And one of the things that scientists often use to distinguish these from one another, if you'll notice, there are sort of these ridges right along the edge of the mouth, that opening right in the center. When the tissue is removed, those ridges are useful in diagnosing which species this cup coral belongs to.
Wide, please, Bob.
And heteropathes. So you saw the proof. I had actually marked in heteropathes
when
tentatively.
When this happens, you might ask
say.
Bridge
so Tina thinks it is.
Azimuthing bow thruster.
Tina has been trying to help me with anophytherian IVs, so I'm glad her time isn't wasted, at least on all of the animals.
Receiver.
Hopefully I won't screw this up, but I think the name relates to the fact that it's got different types of branches on it. There's a cluster of branches in the center of the stem, and then there are longer branches that are out further, thus hetero or different, maybe.
Goes cuckoo, but that's better.
I didn't really want to say that because Tina will probably go,
no.
Yeah, I did.
Okay, I'm going to start tracking.
Because I got it mixed up, but it could be why it's named that way.
You might have to wake up the second one, too.
Oh, I will. You're right.
What is this? Linear feature.
And then in HiPAC, you can go to the vessels, change the track length.
You're reaching.
Yeah,
I'll
you can
clear
just
it out.
you know how to do that.
Linear rock.
Okay.
I guess I'll do that now. Just clean up some of that garbage. Let's see.
Oh, there's a nice little sponge. I wonder if we could get an image of that white globular thing. That's a weird sponge. And there is a beautiful coral after that. It could be another Jasonisis bamboo, but maybe we can get to that after the sponge. This is a weird sponge.
How do you say that in Latin? Obscura?
I don't know. You were being serious. I have no idea.
Well, semi.
Semi-serious.
Bizarro spongia, something like that, maybe.
I think I'm starting to get an idea as he's getting closer. So it's definitely a glass sponge.
Because I haven't gotten
so that's a start. I suspect it's a hard sponge. So that means it's brittle and the spicules are fused together. It's so thin that it's going to be very brittle. And I think it
clear to zoom video.
I think it could possibly be in the family Euretid. And I think it could be Condylasma or in the subfamily Condylasmatinae, but I would sort of throw this out as a possible collection option, but of course I'm biased because I work on these animals, so I certainly don't mind if others think they want to wait a little bit, but what do people think since I'm throwing it out there? It is an unusual sponge. Let's see if we can get some feedback before we move on. And again, if you prefer to wait, that's perfectly fine with me.
So
Tina's good for the collection.
Halfway through that move, if we do want to collect it, we're going to have to stop.
Scott says okay. So I don't know, Pilot, are we in a decent position to try to grab some of this?
Yeah, we can do this.
Nav.
It's going to be very brittle, so expect it to break up when you
present position
when you try to grab it.
Understood.
and I'm sure you already did this, but you woke up number 2, not number...
Yes, I did that.
Okay. Just
we
check.
We just got...
oh, there it is.
And
okay.
it was bad, but we'll give it a couple more.
So, Andy, if you could warm up some hydraulics for me, please.
Hydraulics coming online.
So the first thing we'll do is put a sample ID on here, and so this is going to be D2 dive.
Of course, the box of your choosing.
I guess it's 17
box.
D2.
Oh, yes. Okay.
All available.
I guess it would be dive.
Well, it would be left.
Well, I wonder if you could
port bio box.
See that template? Actually, all you have to do is fill in
over there.
The template.
You might as well get it all shoved out
I'm
there,
not
Andy.
down the button.
I can bring it out
when you get happy and stable with everything.
I can go ahead. You want your port?
Port bio box
Copy
out.
that. So let's retract and then bring it in first.
Okay.
I think there's room for it out there. It might shove the sand a little bit.
So for those who have just tuned in, what we're currently doing is preparing to collect this interesting glass sponge. And so the pilots are preparing the hydraulic arm
I'm looking at
in
45
preparation
right now,
for grabbing and
you might want to bring
putting
it
the
back 5
specimen
meters
into
up.
a collection box.
It'll leave them in a good position
Our
to swing around
co-lead,
and
Chris
observe.
Kelly,
I can always
is
swing around. So.
establishing some of the protocols for collecting off to the side. And we are all getting prepped essentially for the submersible as it begins to collect the sample from the deep sea. So Tina's asking about the zoom on the red thing after the sponge.
So the slant ranges we're getting are right. It's the...
How much more swing do you think I have now? You said minute-wise?
Yeah, probably a minute.
Oh, okay. I just want to keep an eye on that tether.
Absolutely. So on that tracking beacon, we're getting good slant ranges.
Yeah.
It's the the angles that are wrong. I don't know.
The USB on D2, does it respond acoustically or electrically?
It gets electric excitation.
And then acoustically responds, and it's reading it perfectly. So I believe the calibration, it's not the calibration issue, is what I'm saying.
I think so.
Yeah, you have to wait on it. So yeah, you can see how brittle the sponge is. It's always a little bit of a trick collecting them.
Well, we don't need to take the whole thing. We'll just get this little bit over here.
Yeah, that would be great. That'd be more than enough. I know you won't have a chance to try this, but just try this. This is a really excellent job of collecting an extremely difficult animal. So Yeah,
Looks like your
Dave, which
stationary
box is this going to go into?
Maybe so. Yeah, if you think so.
Port inside, and
That's
that's like
port inner, yes.
Yeah, okay. That's it. And you ready to click? Wait till it drops. And it's in the bank, so we can go ahead and
close her up.
Closing up.
You must use this format. Uh-oh. Dave, can you stand by for one moment before you move?
Oh, sure.
We're refamiliarizing ourselves with the SES snapshot. We don't quite have the format right.
I'm just doing a little housekeeping here. I can hang out here now that I can breathe again.
Okay, let's try that. No. Try getting rid of the...
Finished with hydraulics.
Well,
there.
maybe 02, you could be right there. Just add
secure.
02.
Thank you
because
so
only
much.
asking for 2 numbers.
Hydraulic secured.
So what's happening right now, sorry for a brief delay, is that we're attempting to collect all the information regarding the site where this was collected.
Would you like to come in and
That will
fly for a while?
And
Sure,
this is
absolutely.
using
It's a
a
good time
different
to
sort of
switch.
interface here in the computer, and it has a very specific format. Yeah, so it's maybe not parentheses, it looks like brackets. You could try that. We almost got it, but we need to sort of stay here just for a second. They're doing an ROV change as well, so that we don't lose the position of where this animal was collected from. Let's try that. Good grief.
Pilot, I don't think there's anything else I can do to debug this, really. See if it improves when we get shallower.
Copy that. Maybe a problem with the Sirius USBL?
I don't think so, because it's getting the slant range correct, and all the Sirius beacon needs to do is respond to the ping.
It
So
could be multipath.
So for those of you who are just tuning in, this is the Okeanos Explorer, Johnston Atoll Expedition, the Lima Okamoana expedition investigating Johnston Atoll. We're currently on Horizon Guyot, about 1,900 to 2,000 meters, and we're currently in the middle of collecting an unusual and possibly new species of glass sponge from the deep sea. And so we're currently processing and getting ourselves ready for the next step.
Okay. Sorry, just try without the brackets. I don't understand what's wrong, because we're following the format exactly. And if this doesn't work, then we'll go ahead and hand write in the actual data and then figure this out later on so we don't hold up the dive.
Nav, did you drop a target on the collection?
Yes.
Thank you.
Yeah, let's see if this works. Last chance. Hey, we got it. Jeez, always sorry. I'm over 60, folks. I just had an online senior moment, and I apologize for that. But we are ready to move on, pilot, when you're ready. Thank you for your patience, and everybody for their patience.
So this is my colleague, Dr. Chris Kelly from the Hawaii Undersea Research Laboratory.
So I'll
and this is Chris Baugh
back
from
to
the Smithsonian's
340.
National Museum of Natural History. And we're your hosts for the 2017 Naulima Okamoana Okeanos expedition to the Johnston Atoll Unit in the Central Pacific. We're here at 1,900, 2,000 meter depths, looking at a deep sea setting of sponges, sea cucumbers, and other interesting animals present on a manganese a manganese deep sea bottom.
So I wonder, well, we're going to pass it by. We'll let this one go since we're sitting here for a little while. But yeah, we want to see if we can get a few more corals recorded as we go along here.
And Tina had mentioned something reddish, but I didn't see it.
Yeah, I didn't see that either, yeah. Tina, sorry.
Watch lead nav.
Passing through north.
Roger, nav, go ahead.
Unfortunately, we only have a little bit over 30 minutes left on the bottom due to our late start. With that remaining time, should we continue to follow this contour, or would you like us to try to go up over the top?
340.
Let's continue along the edge. I think that's where most people would be interested in us going. Thank you.
Keep doing what we're doing.
So we're gonna continue along this edge. I'm not sure everybody heard that communication with the navigator was inquiring about how we proceed. We have about 30 minutes left in the dive, and we're going to proceed along this edge and hopefully see some other things. Like maybe we could take a look at this coral that's right in front of us with the brittle star in it. If that's possible.
Yeah, I'll come down.
Maybe a coral. Can't tell from this distance.
Yeah, move it.
It has a
corally look to it.
Yeah. I was almost going to say a Chrysogorgia-ish look to it, but...
Co-pilot, should we move ahead?
Yeah, we should. We should get a move going, Gills.
Same as we've been doing, 60 meters at 330.
Yeah. Oh, just back up a little bit, set it down, and we'll take a long look at it.
Because I forgot all about it. We're so busy with this.
Sorry, what was that?
I was telling Andy to back. Oh, sorry.
What was that, Watchley?
I'm very sorry. I pressed the ROV nav instead of Survey 1. Apologize. I was talking to
okay, no problem.
Sorry.
Bridge nav. We'd like another ship move, please. 60 meters, bearing 330 degrees,
what are we looking at with this coral? Is this a corallium? A corallid? I think it could be. Let's give... We're going to need to get a little help from shoreside here as to what we're looking at. Any chance we can get a little closer zoom, or is that not possible? We don't. Yeah. Anywhere here it doesn't have nodes. It's boy, and
that's max now.
Thank you, Bridge.
So, yeah, so I think it's a corallid. I'm not positive. I was kind of waiting to see if others would weigh in on this.
In the meantime, this looks to be a Euryalid type ophiuroid. This is what's called a serpent star relative to the other groups called brittle stars. This group of brittle star or ophiuroids have a fleshy tissue that covers over the vertebrae on their arms, which gives them the ability to coil very tightly on the branches. And these are what's called commensals. They live on these corals. I don't believe they actually feed on them. They simply live on the branches in order to take advantage of water currents and other food swing by them, as well as presumably taking advantage of the protective tentacles, which provide some stinging and feeding for the coral itself. It's not uncommon to find these ophiuroids, these serpent stars, living on corals, and oftentimes the species of serpent star is closely related, or sometimes always related, to the species of coral that it lives on. We'll often see a great deal of these in the deep sea as we progress. This is one of many types. They're fairly difficult to identify, and so there's a lot of work for people who want to pursue research in serpent star, or what's called the Euryalid group of brittle stars.
That's good for Science Pilot whenever video's ready.
Okay, we're out of here.
So I'm going to go with hemicorallium as the best guess on that coral, and since Tina actually also thought it was a coralliid.
Look over to your right until I
so
catch
that's
up
good.
with
We
you.
got a little bit.
Sand to the left.
A couple of us think it's that group.
I'll swing my head over there to keep you in
so,
view. But
Amy
it's a little
Bucco-Taylor,
bit
I
off
suspect,
track.
is not watching right now because it's pretty late for her. So she's really an expert on this group of animals. And
I'm looking 025 right now. Yeah, that's something.
This
to keep
is
us occupied.
Tritopleura. We could probably skip this one.
Well,
We could. Sorry, we've been seeing them all day.
these aren't the droids you want.
That's a live one. We've been seeing a lot of dead ones, but we haven't
Oh, that's
seen as many
interesting.
live
Yeah,
ones.
that's probably the first one live one we've seen today.
Keep eyes peeled for anything red. Red. There was interest in redness earlier.
Looks like another Walteria or a Tritopleura again. I think we've got some images of this guy already. It might be, I don't know if that's a crinoid in the background or a coral. It's to the right, yeah. What is that thing sort of sticking up?
I think it's a stock with some crinoids on it.
Maybe a coral with some crinoids on it. Or is it a stock crinoid? I think it's a stock crinoid. Isn't it?
No, I think that's two feather stars.
Yeah, please.
Yeah.
Oh, darn it. I was hoping to get the echinoderm expert, but he...
No, I got a good eye for it. This is the equivalent. This is the crinoid equivalent of three ducks wearing an overcoat. So. Yeah, continue on.
Yeah, I
Thank
think
you.
we've got some close-ups. Thank you.
So you probably should
It would have been
come around the
interesting
left,
if I was right.
follow the rock
Oh,
trail off
absolutely.
to the
But
northwest.
usually when they're that yellow,
Something in the
the
middle
stock
right
is also
now.
yellow.
Big low sponge. Okay. Oh, I didn't realize how far you were.
Yeah, I'm going to bring...
We haven't seen any sea stars.
you're starting
I mean,
to tug
even
on
though
me
you
here.
specialize in echinoderms, you're very specific.
I'd have to
Yeah.
just come
Here's
back
a
into
here's
the
a new
sand
kind of
a little
crinoid
bit.
on the coral on the right
Back
there.
up.
You notice the white and purple coloration.
Yeah,
I think that's
tug on me there.
one of those telecrinid things with the long
Pilots,
spindly
new video on
arms.
the chair.
Hey, Rolly. Yeah, just keep coming west.
Oh, it's like
Yeah, so
that's that one.
this is new because Chuck just came and weighed in on this and said he's thinking it's a little bit different.
Okay. Oh, it's a oh, geez. Okay.
Yeah.
Xenometridae.
But they've got the long filaments just like the atelocrinids,
Okay.
so it's...
I got to get those straight.
Yeah.
Okay.
Okay.
That was over to the side.
Yeah.
back there.
So as fascinating as this is, you're going to have to take a dash across the sand, and hopefully there's some rocks over on the west side.
think the sea stars are angry at me. We haven't seen one yet.
Yeah, that's kind of unusual. we always see a sea star on every dive.
That said, spatangoids are pretty cool animals, and we rarely see those. So, especially one that's kind of half buried in its habitat like that. One of the great things about Okeanos observations is we often get to see species of animals in their habitat, which is something you don't get to see from my perspective a lot of times, because animals are often collected by trawl net, and thus without the context of how they were collected or what they were doing when they were alive.
And pretty much back on track here. Yeah, looking 340 now. Close enough.
So, Ken Sulak just pointed out that that we haven't seen any shrimp or some of the more common benthic megafauna that we typically see on the dives. They're cruising above the seafloor, or even the nematocarcinus, the long-legged shrimps.
Oh, yeah.
We haven't seen any of these things here, which is...
I thought I saw one swim through just a little while ago.
Oh, did you, Roland? Okay.
Yeah,
okay. I missed that.
What's this?
Yeah, they've got a wispy coral directly going off the screen in the bottom.
And we have...
Bottom center.
It looks like a palmate structure.
Yeah, could you go down? Yeah, there he is at the very bottom. This is something different, I think. Yeah, right there, right by the lasers. I think it could be a Chrysogorgia. Yeah, it is. Yes, it is. So, looks like that's our... I think we saw another Chrysogorgia a little bit earlier, and there's a demasponge next to it, a possible Cladorizid to the left of it as well. So Scott works on these, and he's on the event log. And I'm going to see if he's looking at the animal right now. It could be something that we call Chrysogorgia stellata. It's kind of a bushy member of that genus with pretty thick branches for Chrysogorgids, which this seems to have. But
oh wow!
Yeah.
Is that a little Cladorizid on? Oh
yeah,
wow!
That's what I was saying. It's a little, and he's a little squat lobster. That's what Roland was pointing out on there.
Yeah.
So that kind of waving thing is what we think is the carnivorous sponge. It's right next to the coral.
Andy, you can't see the lobster. It's at the bottom of the screen. Down.
Yeah,
Tilt down a little bit.
Sacco
There
saw
you
a
go.
coral to the right,
Yeah.
too. We'll see if we can spot that afterward. Boys, is that coral planar, or is that more bushy, do you think? It's kind of bushy, right?
The Chrysogorgia?
Yeah. I think it's more bushy. I'm just looking in this on the guide here.
On the stalk, right?
Oh.
Yeah.
Yeah. So there's a sort of a cluster of red spheroid things that almost look like kind of a ball on the coral. They might be eggs, but I'm not sure if they're the eggs of the coral.
Thank you, Bridge.
Ready for full screen?
Yeah, okay, so I'm going to grab that one next.
Little
So you
tilt
know what,
up.
let me see if I can put that in. I think we're good on that. If video's good, thank you.
Video's good.
Okay, you hear these pauses because they're kind of punching information in. Some more dead sponges, one live one, a Walteria we're just passing over. There's a stock crinoid, isn't it?
Yeah, I think it's the same one, though. Another hyocrinus, Tiburana crinus, was that right?
Yeah, I think that's it.
That's a lovely animal.
Right.
There might be some things growing on the stalk. If we could take a quick look, please, at the stalk of the yellow crinoid to the left.
You're good.
Oh, if
good.
Not, don't worry about it.
No, we're fine.
Might be swinging a little bit.
We're not. I'm not moving much. I got you.
Oh, I see. It's a Walteria just near the base. Okay. I thought there was something growing on the stalk.
I'm not.
Oh, and there's a shrimp.
Maxu.
Where's the? I didn't see the shrimp. Oh, it was on the stalk. Oh, okay.
No, it was behind it.
Okay.
Yeah.
Sort of need to get closer if they're wanting to look at that shrimp.
Thank you, pilots.
Yeah, thank you.
Go north, my son.
So you wanna? Were you gonna punch that one in?
Yeah. Oh, sure.
This is another Caulophacus stock sponge that we're going over. We got a good image of that earlier on. This could be bamboo corals. I'm not really sure that we passed over. And we might have something up ahead here on the rock that looks a little bigger. Oh, I see. It's going to be some more crinoids growing on a dead stalk of some kind, I think. Chris?
No, we can move forward. If you see crinoids that are like a strikingly different color, then yes. But if the yellow ones, the yellow feather stars are all that there are, then we can move past them. Thank you. And we appreciate it. Thank you.
Yeah. Oh, yeah, let's go ahead and zoom in on the red. It's going to be another one of these Anthomastus or pseudo-Anthomastus.
Yes.
Let's see if it's a little, I'm not sure if it's different than the other one we imaged.
It looks like it's the same color.
Yeah, the tricky part on them is, one thing from this distance is whether or not the stalk is a different color than the, whatever you call the head of it. Or the,
yeah.
Oh. So it all looks like a uniform color, and
A little tilt down, I can give you more
I'm
zoom.
not sure if Tina's still here.
No, I think she's tuned out for the evening.
Yeah.
It was about 4 a.m. when she logged out.
So I'll take a guess on this one that it's going to be a pseudo-Anthomastus similar to the one that we already had logged. I can punch this one in.
Hold out for full.
So this really shows beautifully the 8 feeding arms with the tentacles coming off of each one. This one is in kind of a mushroom shape with all of the feeding polyps coming off of this large cast sort of structure on the top.
Is that a Bersingiid on the right? Have we seen those?
Sponges on the seafloor there.
Okay.
What Bersingiid?
I saw it, but we moved past it pretty quickly, so no big deal. We'll see another one. So we did actually get to see a starfish today. We just didn't get to stop for it.
So we may have either sponges or corals coming up. I can't really tell from the distance. Be nice again to get some sea stars and get some more corals.
Well, I'm sure we will.
Yeah. Got a few. Yeah, this is another Gidopleura sponge. The live one there, the white one. It's possibly a dead one in the background. I don't think so. I think these are the same things that we've been seeing already today. Thank you.
Nice pirouette would be a nice visual to have of those. Yeah, I don't know how fast we're moving or what our timeline is.
That thing over on the left, is that something we haven't seen before? It's just all squigglied up there. Yeah.
Oh yeah, the squiggly line is a stoloniferous octocoral. We've seen that once before. Chris, do you want to see it again?
But this, not the right under the lasers.
I think we passed it. So, but we saw the stoloniferous octocoral back there.
Yeah.
But what is
so
this?
This is a dead sponge, but it's a different sponge.
Yeah. Okay.
Oh, yeah, we don't need to go back to it, I don't think.
That's dead, isn't it?
So this is dead. The sponges actually turn, these last sponges turn brown like this after they die because they accumulate sediment and they lose a lot of their tissue. But that was an odd one. Interestingly enough, it's not like we were thinking of collecting it, but dead sponges don't make good collections because they lose a lot of the microscopic parts when they die.
It's just off screen bottom. It's
There's something up ahead there.
like
that
something
round,
crawling in the sand in
mound.
the past.
The round mound.
The round mound. Oh, and here's a coral, excuse me, off to the left. I wonder if we could get that just going off screen. Yeah, this one here. This could be another bamboo coral. And then maybe that round thing after this, because that's pretty weird looking.
Oh, and there's a...
Oh, there's another one, a Ritagorgia. Sure, a couple bunch of these. You start with the one on the left there, the close, well, whichever you can get to. Okay, that'd be great.
Oh, on the sponge.
Co-pilot, how are we doing on tether? If we want to move before we recover, it's probably going to have to be about now.
We're good. I'm almost right over him.
Tilt up center.
You see that Les is still on and Scott's still on the event log, and I don't know if we could get a hand with this thing from either one of those, either via the audio or the event log. This is a bamboo coral, something they specialize in.
And let's see the very tip. Hold there. That gives that black in the background.
All right, Les had commented on the other coral, but Les, do you have an opinion on this one? It looks to me like it's another Jason Isis, but I was going to wait for you or Scott to
pull out
comment on
partials.
it.
You can tilt back
This
down and
is
look
the
for
bamboo
any
fan that we saw
additional
earlier in the
creatures on it.
dive. It's got that very opaque stalk to the polyps. I didn't
Very
really
nice.
see where it was branching.
And we'll go full screen from
I
here.
think it's nodal, maybe.
And lasers are clear for the beauty shot.
Oh, I'm sorry, I got, there we go.
A double tap.
Yeah,
And
Maybe
video's
just
clear.
because we're constrained. Yeah, and you know, it's already been identified, the one to the left. I wonder if, given we're time constrained, we could skip this one and maybe go for the blob on the right. Yeah, in the back right. It may be a sponge, but it's... Yeah, it is a sponge, and I think it could be similar to the one we collected. You just do a snap zoom on it just to confirm. Yeah, I think so. I think it's the same
partial
hard sponge that we collected. Great.
Should have lasers
so
on to
Les
go across
Walling wasn't
this.
sure what the bamboo was. Yeah, we're good on that one. Thank you. No, I was just reciting a comment in the event log. The one that we zoomed on just previously, one of the experts was not sure which it was. No, let's go ahead forward, I think. We've got to find one sea star for Chris.
Well, we went past that one Brisingate, but yes, it would be nice. But let's see what else there is.
10 minutes.
Yeah. Well, you can just fiddle around here within the tether length. You can go south on this rock pile.
Looks like it's a crinoid on a dead sponge, I think.
On the right with all the... Yeah, let's take a look. Oh, is that a worm or a fish?
Cucumber?
It's moving pretty quickly.
Oh, there it is.
It's a fish.
It's a fish. I was looking at the rock.
We'll take a look at the rock later, too.
Yeah. Okay, so Ken, you're on. If you can fire off an idea. I have an idea of at least what family this is in.
Yeah, he's...
But Ken Sulak, who was on earlier, let's see if he still is.
He's out.
He's out?
Yeah.
Okay, so this is in the family Synaptobranchidae. So it's a cutthroat eel. And we have encountered them fairly commonly on our dives to these depths and other locations. Okay. Yeah, thank you.
Yeah, let's take... I think there was a rock to the right that had a bunch of spiny projections on it. Yes, sir. Thank you.
Swimming shrimp just went off screen right.
We've got a little bit of bed forms here, indicating there's some current at certain points across the seafloor.
Yeah, just a sort of a quick survey. It looks like sponges and
There's a whip coral.
There's a shrimp to the right.
Sponges.
There's a little red shrimp there, and that's possibly a Candidella, the coral that's off to the right of the rock too. Looks like it's a primnoid. Yeah, another Candidella. Good look at the...
Which one's your interest, Science? Okay, let's go to the shrimp.
Possibly in the genus Plesionica, that would be my guess. I don't know if we've got any arthropod folks here.
Our first shrimp today. And when we're done to the far left of the rock, there was something in the sand that might be a jellyfish. We're
Video's
not
clear.
Sure. Awesome. Thank you.
That little pop on the camera just then was a loss of communication momentarily. I think you got a sponge there. I think that's it.
Okay, yeah, let's take a look at this.
It might be a sponge.
Yeah, nothing wrong with that.
No? No, I think Dougal was hoping it was a jellyfish.
Oh, okay. Actually, this is an interesting sponge here. We could get it's different
a little
than the
bit more
ones we've
rotate
seen
right so you
earlier.
can see the
It's living in
stalk.
sediment. That's one clue.
I think they're going to be interested in the stalk structure.
We'll get
as well.
a quick zoom because I think there's an effort to get some jellyfish and sea stars. Yeah, so this particular sponge is in the family Pheronematidae, and it's in a different genus than those big massive ones. This is called Sericalophus, a catcher's mitt sponge. So we're kind of seeing it from a side view, but if you looked at it from the front view, which is facing down, it would look like a catcher's mitt. So they're really very cool sponge. This is a deep water version, and we're calling it, I think, Sericolophus new species 1.
Oh wow! So again, for people who are just tuning in, this is a glass sponge, and one of the neat things about this, as we're seeing very closely, is that this holdfast, this root, if you will, these are all glass fibers, and so these are very similar to the fiber optic cables that might be connected to some of the electronics in your house.
And
the texture and the feel is very similar, and in fact, there are industrial people who are really interested in how sponges grow these glass fibers in order to build better fiber optics.
Okay, we're clear.
Thank you.
3 minutes left.
If you come left and go west, you'll be setting yourself up for recovery a little bit.
Add a minute or two if you're going to get set up ahead of time.
Is that another Brisingid there going off screen left? Wrapped around the sponge.
So there's a white star-shaped animal on those two central stalks, right under the crinoids. I think that's probably a Brisingid sea star of my dreams.
Well, better get that in the bag.
We got a sea star. All right, woohoo!
So this is a Brisingid sea star. These are very similar in appearance to brittle stars and feather stars. You'll see all of those long projections coming off of the arms. Those are used. So this animal's natural feeding position is to raise all of its arms into the water, and it uses those spines to capture food as water currents bring tiny crustaceans near them. The spines have little claws on them called pedicelaria, and those little claws completely cover the spines, and they form almost kind of a death Velcro. And so when small amphipods and little tiny crustaceans swim by them, they get caught on them. And then the tube feet clear off that food and move them down to the mouth. This one looks like it might be in the genus
top under the arm,
maybe Fraella. These are commonly, this particular group is commonly encountered at deeper depths.
You have the ability to rotate right
surface
without
of the arm plating is usually more sort of consistent. Other groups have more well-defined ribs. But this one looks like it's kind of passively resting. It's got its arms down instead of extended into the water column. But first good capture of a sea star today, and I'm very grateful to everyone for spotting it and zooming in on it.
Got a black coral in the background too, probably another one of those potential Chrysopathes, which Tina helped us with earlier on.
Going for full screen.
As we're getting close to leaving the bottom, that glass sponge is genus Tritopleura. That's the one to the right of it.
There's a little sort of translucent crustacean on that upper arm right in the middle top. I'm not sure if it's I'm not sure if it's just caught or if it's just hiding below the spine.
Hold. Chris, it's actually sitting on the sponge behind it.
Oh, it's on the sponge behind it. Oh well. Still kind of interesting gold eyes, though. Yeah, sometimes the crustaceans are just caught on the spines. Like that one might be down to the right of it.
By the way, we wanted to give everybody a heads up that we are going to have a dive planning call this afternoon right after the ROV leaves the bottom.
Okay, so basically if I just swing around right
there,
to
we know it's pretty late for some of you. So if you
swing
can
around
a
right
few
to
minutes
the west
you want
and
to
drive off,
participate,
we should be good.
With regard to tomorrow's
I got
dive.
your swing arms. double check
be a full-length dive.
I think I got them.
As far as I know, but we'll get an update from the ROV dive
pulling
super
up, give you
during the meeting.
Clearance here, and coming around west. Push out until it looks good. See the tethers starting to come around.
What is that big? That's just a rock, Shadows. The rocks can really play funny things with your mind down there. You can see all types of shapes in them.
Yes, well, I've seen any number of quote-unquote sand stars, starfish-shaped bodies of sand amongst the basalt or the manganese.
Look at that ring of holes we're coming up on.
What's that interesting sort of
I've seen
sediment
that
structure
before?
That this little thing right here?
Yeah, the looks like a bunch of round dots in a circular pattern.
I think they're getting ready to leave the bottom. If I'm
You're
not
getting
mistaken.
stretched out. You should be able to start coming up.
I think it's more than just getting ready. I think
We have left.
they're fully lined up now.
Elvis has left the building, huh? Okay.
Very good. While we're still sort of on the bottom, is there time to let the pilots introduce themselves? We haven't had a chance to do that today.
Sure, it's
Camera up front.
Yep.
co-pilot Dave Wright.
Navigator Dave Casagrande.
Yeah, hiding in the corner is Roland Bryant.
Tyra Smithy at Video Clipping Station.
So
Picking you
thanks,
up in
guys.
tether cam. Keep pushing, keep
thanks
rising.
For,
Bridge nav.
thanks
also-
to the ship for actually troubleshooting
left
that
the bottom,
problem
and
and
we'd
actually
like to
getting us a
continue to
dive
hold
today.
position
It was
for
looking
now,
pretty
but,
shaky there for a while.
I'd also like to thank Nicola Rodriguez, who's helping us annotate and working on
Okay, tether's all good. Starting to haul up.
and working helping us annotate a lot of the animals that we're seeing.
Yeah, so again, real quick reminder, as soon as we can get a hold of Mushcore, who's probably in the room next door, then we're going to go ahead and have a dive planning meeting. So give me just 5 minutes to round him up and give him time to bring up the Fleeter Mouse maps that we'll use to show you what we have planned for tomorrow, and we'll get right back to you.